everybody, and welcome to today's photosynthesis webinar. It's great to be back. I'm Louisa Diva, and these webinars are funded through the Capitalise project. I'm delighted to introduce our two guest speakers today, Dr Joana Amaral from Lancaster University. Joana is a postdoctoral researcher at Lancaster University in Elizabeth de Calma Silva's lab. Her research focuses on understanding plant responses to their surrounding environment and enhancing plant performance under different scenarios. She is also part of the Capitalise project, studying strategies to improve photosynthesis for higher yielding crops based on rubisco regulation by rubisco activase. And today she's going to talk to us about regulation of rubisco activase activity in shade-sun transitions.
So thank you for the introduction, Lisa. Can you see my slides?
Yes, great.
Yeah. Okay. Today I'll talk about rubisco regulation in response to different light levels and how rubisco activase is a key player in this regulation. My slides are not moving. Oh, okay. I'll start by presenting the project itself. So Capitalise focuses on improving photosynthesis for higher yielding crops. It does so by exploring the natural variation in important crops in Europe, and also does some GM work, mainly to try to identify the mechanisms that are behind photosynthetic limitations. There are three main approaches that the project focuses on. One is tuning leaf chlorophyll content, and I will not go into detail into this one, and you can find more information about it in the Capitalise website. There's also the improving calving cycle, and we, as you can imagine, focus on this part of the project. There are also other groups focusing on RUBP regeneration, but here at Lancaster we study more rubisco and how it fixes CO2. And the last approach would be focusing on improving reactions to light, so photoprotection mechanisms, and also how rubisco regulation happens in this response to changes in light. So we consider three main crops for this project: barley, maize, and tomato. Different types of photosynthesis and monocots and dicots. And what we have available is several germplasm collections that we can work with, and there has been a great effort to phenotype these collections, genotype them, and then find markers that could be useful for crop breeding. But also from this work we could get some interesting genotypes or lines that have contrasting responses in terms of photosynthetic capacity, and that could be useful also for this deep phenotyping. And here we would be more interesting on studying the rubisco properties of these plants. So now going into rubisco regulation itself, here you can see a network of factors that can impact its activity. You have, for example, an activator, carbon dioxide or magnesium, that are essential for its activity to make it, that are required for rubisco to work. But the most interesting or important factor would be rubisco activase, which is its chaperone, and as its name says, it's responsible to keep rubisco active, mainly because of it releases inhibitory sugar phosphates that bind to rubisco and inhibit it. And rubisco activase is quite diverse. There are different isoforms. Its abundance is different in different species, and these different isoforms also respond in different ways to temperature and light. So, for example, you would have in Arabidopsis a single gene that through alternative splicing produces a beta isoform and an alpha isoform. So the beta is a shorter isoform, about 43 kilodaltons, and then the alpha, it's a longer isoform of about 46 kilodaltons that has this C-terminal extension where you can find two cysteine residues that allow for redox regulation. In tobacco, you would have only beta isoforms, and in wheat, there's one gene coding for the one beta isoform, and then another one that by alternative splicing again produces a beta and an alpha isoform. And so Rubisco is inhibited in the dark and under low light. So plants in the field, they are exposed many times to transitions in shade to sun. And the problem is that Rubisco reactivation could be slow, and that reduces the potential for CO2 assimilation. And estimations, for example, in wheat say that this could be by 20%. And again, Rubisco activase has an important role in this because it removes inhibitors, so interacts with Rubisco, changes its conformation, and allows for the release of these inhibitory sugar phosphates that bind to it in the dark or under low light. And it does so in an ATP-dependent manner. Rubisco activase is regulated by the chloroplast redox status, ADP-ATP ratios, and is also sensitive to changes in temperature. So as I just told you, these two different types of RCA isoforms differ in that sensitivity to redox regulation. So in the alpha isoform with that C-terminal extension would be redox sensitive, and in the dark would be oxidized. So there's these disulfide bonds here and would become more sensitive to inhibition by ATP. And this comes from studies in vitro, initially in Arabidopsis, where they found this difference between the alpha and the beta isoform. And by using deletion mutants, where they would remove pieces of the protein, 7 amino acids and 14 and so on, they found that ADP sensitivity was connected to the terminal 7 amino acids, the last 7 amino acids. And then they produced some site-directed mutants of this protein, and they tested how they respond to changes in ADP-ATP ratios. So in the light, it would be similar to 0.33 ADP-ATP ratio, and in the dark, we would have a 1 to ADP-ATP ratio. And what they've seen is, so the beta isoform has some sensitivity to increases in ADP, but the alpha is the one that really, as soon as it finds some ADP, it gets inhibited. And when we do these mutations for the cysteine residues here, what we find is that the decrease in activity is not as strong as seen for the alpha isoform. And so this tells us that the cysteine residues are actually essential for greater ADP sensitivity of this alpha isoform. And as the cysteine residues could form these disulfide bonds, then they tested how thioredoxins could be involved in this process. So you would have the alpha isoform going down here, but once you incubate it with thioredoxin F in a reducing environment, this is not true. So activity is higher. And again, Rubisco activity goes up in these conditions over time. But if, again, you allow it for this cycle of reduction and then oxidation, activity goes down again on its oxidized environment. So it seems that RCA alpha is reversibly activated by this reduction specific to thioredoxin F. These authors also tested how alpha and beta isoforms could interact. So as you can see, beta isoforms have high activity if you put them together, and this is in a 1-to-1 proportion, 50-50, what is found in nature in Arabidopsis, activity goes down. A reduction cycle with the thioredoxin F increases activity, and then this is reversed by, again, another oxidation cycle. So yeah, it seems that redox changes in RCA alpha via this regulation by thioredoxin can regulate actually both isoforms, as they tend to work in a complex. Also, this oxidized form of RCA alpha showed to have lower ATP affinity than the reduced form, or then even the beta isoform. Then when moving to plants and having plants expressing both the beta and the alpha isoform, only the beta, only the alpha, or no RCA, you can see that plants that do not express RCA, they do show limitations in CO2 assimilation rate, as well as plants expressing only the alpha isoform. Some experiments were made with growing plants under high light and then having them under low light for 1 hour and then allow them to recover under high light. Again, plants with no RCA struggle here with Rubisco activation. And then we have two main profiles. So wild types, plants showing both beta and alpha, plants showing alpha, and again, alpha and beta. They show a decrease during incubation at low light, and then a slow increase when they go back into high light. While plants that express only the beta isoform or mutated forms of the alpha in their cysteine residues are able to maintain Rubisco activation always at high levels. So this seems promising, and from these results, there's the question if we can actually improve photosynthesis when we remove this redox regulation of Rubisco activase. Other studies also considered Arabidopsis and tobacco. So in Arabidopsis, again, species transgenic lines expressing only the beta isoform do not show any sensitivity to ADP, ATP ratios increase. And tobacco, which only expresses beta isoforms, was interesting to see that it shows some sensitivity to increases in the ADP-ATP ratio. And some mutations were made for this tobacco beta isoform, and there was no improvement except when you actually changed it for the 17 amino acids that are present in the beta isoform of Arabidopsis. But what was more interesting in this work was that the plants expressing only the beta isoform, again, in these plants, Rubisco was not deactivated at low irradiance. There was a faster induction of CO2 assimilation after transition from low to high light. But what was interesting is that the growth, plant growth was enhanced under fluctuating light regimes. So wild types were showing some less biomass when grown under fluctuating light regime, and transformants actually could keep their growth. And then we moved from model species into crops. So as I told you before, wheat has 3 isoforms, and all of them showed sensitivity to ADP inhibition if we look at the activity of ATPase hydrolysis from RCA. But if we look at actually how much it reactivates Rubisco, one of the beta isoforms only one of the beta isoforms shows to have some resistance to this increase in ADP-ATP ratios. So then some site-directed mutants were made by comparing the sequences of these 2 beta with the 1 beta that showed different responses to ADP. And what we've seen is that when changes occur in the C domain, there is a decrease in the activity when they find ADP. So it seems that ADP sensitivity is associated to the C domain. And this was interesting to see that with only small changes, we could basically change the properties of RCA. A more recent study showed that if you reduce the RCA alpha content in soybean, and they did experiments under controlled conditions and in the field, there was no impact on the morphology, leaf expansion rate, or the total biomass of these plants. There was no change in the photosynthetic induction rates. Plants maintain the ability to regulate Rubisco activity in low light. And so it seems that in this species, RCA alpha has not the same impact as it has in Arabidopsis, as we've seen. And this could be related to their basal content. So in soybean, we have about 10% of RCA alpha, while in Arabidopsis, we have about 50%. So this study actually tells us a lot of things that we have to keep in mind when we are studying these approaches. One is that we need species-specific solutions, and we need to understand Rubisco regulation in the target crop that we are working with. And also that it is important to report these negative results so that we can learn how to explore these strategies. So, bearing all this in mind, we are now developing our own studies in tomato, trying to understand how RCA is regulated by light in this species. And we chose this species because it only has beta isoforms as it is seen in tobacco. And it was quite interesting to see how it is inhibited by ADP, increasing ADP-ATP ratios. I will not tell you much about the results that we have so far. It's mostly focused on the approach because we still don't have a full story for this. But basically, we have an in vivo part of this project where we are growing plants under high light and under low light or limiting light. Then we incubate our some leaf discs from this plant in our light rig system, and again under high light or low light. And then we evaluate Rubisco and Rubisco activase activity and amount, and we explore the redox status of RCA. This is just to show you the light rig system we have here that has some LED lights that mimic clear sky solar irradiance, and we can control light levels and also temperature. And by using this system, we make sure that what we are seeing in our results is actually what was. We avoid any shading or anything. We can then complement this with in vitro studies. So we purified these isoforms from an E. coli expression system, and then we are doing tests like similar to what I was showing before, to check if these isoforms are redox regulated or if they present some sensitivity to ADP. And of course that we have to do also some in silico work where we compare the sequences from these isoforms in tomato, but also with other species that are better studied. And I hope I can show you some results soon from this work. But for now, I hope that you got the idea that RCA has potential to increase photosynthesis under fluctuating lights, and that we need to search for species-specific solutions. So, as we've seen that one size does not fit all, we need to understand RCA properties in the target species, and that means check the abundance of these RCA isoforms and also how they respond to challenging scenarios, and also understand the regulatory mechanisms that are behind this. I just want to take this opportunity to show you the people that are working in the Capitalize project, so the Capitalize consortium, but also the team at Lancaster, the photosynthesis team, which are the people I work with every day. And yeah, so this is the end of my presentation. Hope you enjoyed it, and open to any questions. Thank you.
Thank you, Joanna. That was really interesting, and I have lots of questions myself, actually. Really fascinating. But if anybody else has questions, please put them in the question box, or I will hog question time. Okay, my first question is there an optimal ratio there between your alpha and betas? And is that ratio, then you could choose a ratio for each environment, for example, whether it's fluctuating light or heat or whatever. Is there an optimal, or is it potentially you could find an optimal ratio?
I don't think at this point we already know an optimal ratio. I think the idea would be to select for, okay, if you see some interesting response to light or to temperature in a specific isoform, you would select ideally for a plant that expresses more of that isoform. But I don't think we are there yet, because as I was saying, even for most species, we actually don't know their basal levels or how they respond to changes in the environment or in the chloroplast environment.
So do you think it is fixed in a species, or do you think it can change then? Do you think if they're grown in different environments, would you expect it to change?
Yeah, I think we've seen before that the expression of some isoforms could be, for example, under heat, it could change the balance you have from different isoforms. So you could have some isoform that it's actually not a big part of the pool of RCA in optimal conditions, but then when the plant goes into some stressful condition, you find that it increases its abundance. So it could also be related to some stress response.
Yeah, really interesting. What's my other questions? Let me see. I was thinking about sort of whether you could, for example, if you did know what the optimal ratio was, it'd be a really nice screen to, you know, if you're looking for plants that are better adapted to a particular environment or crops, you could pick varieties that had a particular ratio in those. Was my thought as a screen thing, which is probably simplifying it incredibly.
Yeah.
But
No, I mean, we could do many things in vitro, of course, testing, like, okay, if I have this ratio of isoforms, like how does that change Rubisco reactivation? But then we know that things are a bit more complex in planta. So we have to think about this too. But yeah, maybe screening collections and find if there is, like, in plants that perform better, for example, under heat, do they have a different pool of RCA?
yeah,
that could be interesting.
yeah. And I mean, there seems to be a lot of potential improvements to Rubisco activity because it is so, in many ways, so flawed. Have you got a favourite strategy you think is the way forward, or are you fully supportive of your? Do you think it has to be a series of multiple strategies?
Yeah. So as you've seen, the first slide I showed this network, and it's really complex, like Rubisco regulation. And I think we have to start somewhere. And Rubisco activase, as we showed it also in that figure, seems to be a central player in this regulation. So probably if we can improve its response, it would keep Rubisco active. And I think it's probably our best shot now. But yeah, then we might need to add then other layers of improvement to that.
Yeah, because one of them, I think I'd read or you'd said that if you only have one of the isoforms, Rubisco is always active. Is that right? And is there a downside to being active
with the beta isoform? Yeah, it showed that in Arabidopsis. where there is this pool of 50% of alpha, 50% of beta, and it seems to work on that species. But then if you look again at soybean, which only has the 10% of the beta, things are not quite the same, of the alpha, sorry.
Yeah. So is there a downside to always being active if you're for Rubisco? Is it, or is it just an energy?
It doesn't. It seemed to work fine in one of the works with from Elizabeth with Arabidopsis. We could actually see better an increase in the biomass, but yeah, it's yet to be tested in crops.
Mm-hmm. I've got a question just come through. Great talk. What do you think could make the different number of isoforms or different activity depending on plant species? What do you think could make the different number of isoforms or different activity depending on plant species? Are the plants that have more RCA, are the plants have more RCA isoforms or activity relatively tolerant to stress?
So yeah, during evolution, you could see that there are different isoforms, and some species have many isoforms while others have only one or two. And I'm not sure if there is a link already with having more or less different types of or different isoforms would lead to more or less tolerance to any stress. I'm not sure if I'm answering the question, but at this point, yeah, I'm not sure if having more options would allow plants to better adapt to a certain stress or not.
Okay then. Well, if there's no more questions, then I'm going to wind us up here. And thank you both for two really exciting talks. I enjoyed them both really well. And thank you to the audience for the discussion and the questions. I'd like to remind you that our next month's webinar is on the 5th of December with Professor Zoran Nikolowski, and he's talking about machine learning of photosynthesis related traits using hyperspec data. Professor Paolo Pesaresi, who is the leader of a new EU photosynthesis project called Best Crop, where they're boosting photosynthesis to deliver novel crops for the circular bioeconomy. So both really interesting speakers. So the registration is open for that now. Yes, and that just leaves me to thank you for joining us and thank our speakers once more, and hopefully see you again next month. Goodbye. Thank you.